So today I am going to discuss about the industrial production of penicillins, and it is given in your syllabus. Especially the penicillins is mentioned in your syllabus. So let's get started now. So about the penicillins, penicillins are a group of beta lactam containing bactericidal antibiotics. That means the penicillins can kill off bacterial cells which are pathogenic. That's why it's naming bactericidal. Better to say bactericidal. Cidal means to kill bacteria. Now, being the first among the antibiotics to be discovered, penicillins are historically important because these penicillin are actually the first antibiotics which has been discovered by Alexander Fleming. And the structure of important penicillins, some are synthetic penicillin, some are semi-synthetic penicillin, has been given on figure which is given on your side. But the basic structure of all the penicillins consists of a beta lactam ring. It consists of a lactam ring and a thiazolidine ring. So there is a lactam ring present and a thiazolidine ring which has been fused together to form penicillin known as 6-amino penicillinic acid or 6-APA, right? So general structure of penicillin has a beta lactam ring, it has a thiazolidine ring which are fused together and forms 6-amino penicillinic acid. Now there are different types of penicillins are there, semi-synthetic, synthetic penicillins are there, and all have some common features. They have this dialactam ring present, they have this thiazolidine ring present. The difference is only what is present in the R group. So the R group will vary among the different semi-synthetic penicillins that we will see. So here is a list of R groups which has been placed here. I'm minimizing here. So you can see here all the R groups has been mentioned here, and on the basis of the R groups there are different types of penicillins. All have these two things in common. They have this beta lactam ring, they have this thiazolidine ring, right? So let us see some of the semi-synthetic, some of the biosynthetic and semi-synthetic penicillins. The most common penicillins that we will find is your penicillin G. Okay, you will find them as penicillin G. Penicillin G is also known as benzyl penicillin because of the presence of this benzene group attached with a methyl group. You can see here. Another biosynthetic penicillin is your penicillin V, having this substitute, having this group present in the R group of 6-amino penicillinic acid. Some semi-synthetic penicillins, as we know, is ampicillin. This is the R group which is present in the ampicillin structure. Okay, then amoxicillin that we normally eat as antibiotic when we have a lungs infection, amoxicillin. Then oxacillin, look at the structure of oxacillin, the R group, only the R group has been mentioned here, and the rest of the structure is your this one. Okay, the rest of the structure is this one. We are mentioning only the R group below. Then comes cloxacillin. Look at the structure of the R group that is present in the cloxacillin, a different type of semi-synthetic penicillin, which are all produced during manufacture process. Floxacillin, then ticarcillin. Okay, so many and these are some important semi-synthetic penicillins which has been mentioned. There are more than that also, okay, which are being prepared. So the action of penicillins, what it is written here, Natural penicillins are your penicillin V and G. That's why they are called as biosynthetic penicillins. So natural penicillins are effective against several gram positive bacteria. Now we name them gram positive and gram negative depending on the constituents present in their cell wall. Okay, gram positive bacteria and gram negative bacteria have little bit structural difference in their cell wall arrangement. Okay, and by the help of gram staining technique as discovered by Christian Gram, we name them as gram positive and gram negative bacteria. So, natural penicillins are effective against several gram positive bacteria. Normally, natural penicillins inhibit the bacterial cell wall synthesis, which is your peptidoglycan synthesis. So, there is an important component found in the bacterial cell wall, and that important component is your peptidoglycan. Now, natural penicillins normally inhibit this bacterial cell wall, inhibit this peptidoglycan synthesis, thereby hampering the structure of the cell wall. So, the bacterial cell will eventually, due to osmotic lysis, due to the excessive pressure inside the cell, it cannot able to adjust the osmotic pressure and cause cell death. Now, there are some persons, approximately 0.5 to 2% of a population, are allergic to penicillin. Now, this may occur because of allergic reactions, hypersensitivity reactions that may occur in some allergic and in some individuals. That means they are, in that case, the penicillin is acting as an allergen, as an antigen to trigger the allergic reactions. Natural penicillins are ineffective against those microorganisms that produce beta lactamase. So there are some microorganisms that produce beta lactamase enzyme that can disrupt your lactam ring found in the penicillin. That's why natural penicillin is ineffective against those microorganisms which produce beta lactamase enzyme, because this beta lactamase enzyme will hydrolyze the penicillin by breaking that beta lactam ring found in the penicillins. Like for example, Staphylococcus aureus. Staphylococcus aureus, they can produce this beta lactamase enzyme, and that's why natural penicillins are ineffective against this microorganism. So what to do? Therefore, we prepared several semi-synthetic penicillins which are resistant to beta lactamase hydrolysis, and thus several semi-synthetic penicillins are being developed, and they are successfully used in large number of gram-negative bacteria. Okay, like for example, I have said the name ampicillin, then floxacillin, then azlocillin, then cloxacillin, then amoxicillin. These are some examples of semi-synthetic penicillins which are effective against those microbes which produce beta lactamase, because natural penicillins will get hydrolyzed if they come contact with these microbes, because they have this enzyme exposed, and this enzyme is normally degrading the hydrolyzing the beta lactam ring in penicillin. That's why we have to prepare semi synthetic penicillin like this in order to resist against that beta lactamase forming microbes. Okay, from huge quantities of penicillin produced by fermentation. Okay, normally penicillin can be produced by the fermentation reaction, the same fermentation process that we used for preparation of alcohol. In the previous lecture, we have said about the alcohol production process by fermentation technique. Similarly, here also we will produce huge quantities of penicillin by fermentation through submerged culture. Through submerged culture. Now, what is submerged culture? Here we will use liquid medium, okay, so that the penicillins get dissolved in that, the penicillins get suspended in that liquid medium, and will trigger, we will give add the nutrients for growth of the penicillins, for growth of that microorganism, the fungus from where penicillin can be obtained actually. Now generally the fungus which was actually discovered by Alexander Fleming that can synthesize penicillin was Penicillium notatum. So Penicillium notatum was actually uh, mentioned that this fungus can able to prepare penicillin. But nowadays, for commercial production of penicillins, semi-synthetic penicillins, we go for Penicillium chrysogenum. We go for Penicillium chrysogenum. CHRY chrysogenum. Okay. So normally now we use Penicillium chrysogenum for the production of penicillin. Now, and all this process is done by fermentation process. So we go for fermentation technique, and we do submerged fermentation. That means we use liquid medium, liquid broth culture for that contain all the nutrients required for growth of this Penicillium chrysogenum for the synthesis of the penicillin. And thus overall production process, what we recover, 40% of the penicillins are used for human healthcare, 15% of the penicillins used for the animal healthcare. Okay, veterinary science. Okay, and or you can say animal husbandry. And 45% of the penicillins are prepared for semi-synthetic penicillin production. More research is going on. Okay, so penicillin have quite very importance in nowadays also, especially when we are infected with this coronavirus infection. We have scientists have tried different combination of antibiotics to actually stabilize the human body when they are getting exposed to coronavirus infection. And one of the antibiotics is also penicillin, and a semi-synthetic penicillin known as amoxicillin. Okay, which is quite effective. Other antibiotics were also being discussed, like azithromycin. Then your um, other antibiotics are also being discussed in statistics has been made that when treating with patients with this type of antibiotics, what are the number of patients which are recovering quickly from those infection. So that's another issue, but it is being in this much we can understand that penicillin production we need a lot because it has very importance in your human healthcare facility. Okay, now in earlier days, Penicillium notatum was used for large scale production of penicillin, as discovered by Alexander Fleming, that this fungus can be used for large scale production of penicillin. But currently, Penicillium chrysogenum and its improved mutant strains are preferred. What does mutant strains means? Mutant strains means that we use this Penicillium chrysogenum and we try to do mutation. We try to do mutation. What does mutation mean? Well, mutation means change in the base pair of your DNA. Okay, change in the base pair of your nucleotides found in the DNA. Now we all I think you know about the nucleotides. You have studied little bit about in your class 12. Nucleotides are the building blocks of DNA. Nucleotides are the building blocks of DNA. So many nucleotides make your DNA and RNA as well. So by changing this nucleotide sequence, uh, how we can change the nuclear sequence by some physical agents like UV rays. We can use UV rays, we can use X rays, okay, or we can use some chemicals like ethidium bromide, ETBr. We can use some more chemicals, okay, to induce mutation, to induce change in the base pair of DNA, so that by changing the base pair, maybe it is possible that that particular after changing that nucleotide sequences, maybe that particular genome where the nucleotide sequence has been changed by doing mutation, maybe it has some beneficial characteristics. So some mutant strains, some mutant strains of Penicillium chrysogenum has been prepared by causing mutation, by causing mutagenesis. That means we create mutation within the genome of Penicillium chrysogenum, and to make improved variety of Penicillium chrysogenum, which can be employed for large scale production of penicillin. Right? I think you have understood. I have just little bit in short, I have tried to say about mutant strains means what. We are mutating the nucleotide sequence within the Penicillium chrysogenum here, so that to make a improved quality of Penicillium chrysogenum, right? And that has a more capability of production of penicillin during fermentation process. Next, it is saying previously the penicillin production used to be less than 2 units per ml. So presently, sorry, previously, when we are producing the penicillin, it is saying that less than 2 units per ml of penicillin was being produced during fermentation process. But nowadays, after creating this type of mutant strains of Penicillium chrysogenum through mutagenesis, so with the new strains, production runs into several thousand of units per ml. Now this mutagenesis is purely little bit of by chance, because there may occur that mutant, that the mutation that we have done in Penicillium chrysogenum may be beneficial or may not be beneficial. Okay, so by random mutagenesis, by randomly doing mutation at several sites of DNA within this Penicillium chrysogenum genome, we have created such mutated Penicillium chrysogenum strains that can able to produce several thousands of penicillin units per ml of the culture medium. And one of the high yielding strain is your Q176. So Q176 is a what type of strain? It is mutated strain. It is a mutated strain of Penicillium chrysogenum, which produce several high yielding capability, which has several high yielding capacity of penicillin production during fermentation process. So this is another chapter actually which will be given in your sixth semester, maybe in your sixth semester, mutagenesis. Okay, there are different types of mutation that we can go. We can do random mutagenesis. We can do PCR based mutagenesis by applying mutation, by applying change in the base pair of the DNA. Okay. So Q176 is an important mutant strain obtained by mutagenesis of Penicillium chrysogenum, which can yield high amount of penicillin per ml of your culture medium. Now, genetic engineering for improved penicillin production. Some of the genes involved in penicillin biosynthesis by Penicillium chrysogenum has been identified. So the genes which are involved for penicillin biosynthesis within the genome of Penicillium chrysogenum has been identified. Thus, genetic manipulation were carried out so as to substantially increase the penicillin production. It just means to say that we have analyzed, scientists have analyzed the whole Penicillium chrysogenum genome. They have sequenced the whole Penicillium chrysogenum genome, and they have identified that what are the genes. Genes are, you know, essential segments of DNA that are capable to code for protein, for enzymes needed for your cellular process to occur. So they have identified what are the genes, what are the enzymes responsible for the penicillin production. And by studying those genes, we can go for genetic engineering. That means we can modify the genes with the help of certain tools, like we can use restriction enzyme to cleave, to cut a certain region in the genome, to introduce our gene of interest having more capability of penicillin production, maybe. So these some tools or techniques can be employed to regulate the synthesis of penicillin and increase the penicillin production. So through genetic engineering, this much you remember, that through genetic engineering we can able to carry out more modified process to increase the penicillin production. For example, extra genes coding for enzymes cyclase and acetyl transferase have been inserted into Penicillium chrysogenum. Okay, why why extra genes encoding for enzyme cyclase, genes responsible for coding acetyl transferase has been introduced to the Penicillium chrysogenum? Maybe this cyclase enzyme, acetyl transferase in sorry acyl transferase enzyme, maybe they are involving in penicillin production. Okay, so by observing the genes of Penicillium chrysogenum, we scientists have identified those sequences which are those genes which are responsible for penicillin production, and by genetic engineering technique we can manipulate that production, we can increase the production by many way. Like for example, we can introduce the genes, we can introduce the extra genes that code for enzyme cyclase, acyl transferase. Why? To increase the penicillin production. Now during the pathway, it depends on the pathway actually. When we will see the biosynthetic pathway of penicillin, then we can understand that why we are adding cyclase extra gene for cyclase or acyl transferase. Let us see the biosynthesis of penicillin now. So if you see the biosynthesis of penicillin, I will go for the chart here, the flow chart that has been given here. Let us see. So you can see here a name of a acid known as L-alpha amino adipic acid. So L-alpha amino adipic acid is the precursor molecule. It is the initiator molecule for your penicillin production, which is found inside the genome of your Penicillium chrysogenum. So L-alpha amino adipic acid, it combines with L-cysteine and L-valine. It combines with L-cysteine and L-valine to form a tripeptide known as L-alpha amino adipyl cysteine valine. So you can see in the first step, L-alpha amino adipic acid combined with L-cysteine, which is an what? It is a amino acid. It is a amino acid, right? This L-cysteine and L-valine both are amino acids, right? So it combines with L-cysteine to form L-alpha amino adipyl cysteine, and later on L-alpha amino adipyl cysteine combines with valine to form a tripeptide, and the name of the tripeptide is alpha L amino adipyl cysteine valine. Alpha L amino adipyl cysteine valine, which is a tripeptide. Okay, tri means made up of three, peptide means there is a peptide linkage that joins these amino acids, right? So this is the first step that we can see in the penicillin production. Okay, Nextly, this compound A alpha L amino adenyl cysteine valine, so these tripeptide will undergo cyclization. It will undergo cyclization to form isopenicillin by the action of enzyme cyclase. And that's why we have said that if we introduce the enzyme cyclase by introduction of the gene that code for cyclase, so if we introduce the gene responsible for coding the cyclase enzyme is introduced here. So cyclase, what it does, normally cyclase convert this tripeptide known as L alpha amino adenyl cysteine valine to be converted into isopenicillin N. So it is converted into isopenicillin N, and this isopenicillin N then reacts with phenyl acetyl coenzyme A. You can see here phenyl acetyl coenzyme A as isopenicillin is reacting with phenyl acetyl coenzyme A to form penicillin G. So again I repeat, isopenicillin that has been produced in the previous reaction done by the cyclase, this isopenicillin now react with phenyl acetyl coenzyme A and form penicillin G. Well, in this step, this reaction will be catalyzed by this enzyme acetyl transferase. Sorry, acyl transferase. I am repeatedly saying wrong word. Acyl transferase, ACYL, acyl transferase. So normally we can see that so in the first step, let us repeat this step again. So L alpha amino adipic acid combines with L cysteine, and nextly it combines with L valine to form a compound, to form a tripeptide called L alpha amino adipic cysteine valine. Well, you should write L first, then alpha. L alpha amino adipic cysteine valine. Now, this L alpha amino adipic cysteine valine then undergo cyclization catalyzed by this enzyme cyclase to form isopenicillin N. So L alpha amino adipic cysteine valine undergoes cyclization catalyzed by this enzyme cyclase to form isopenicillin N. Isopenicillin N then react with phenyl acetyl coenzyme A in the presence of this enzyme acyl transferase to form penicillin G. Normally, what does acyl transferase do? Acyl transferase exchange the amino adipic acid with the phenyl acetic acid. So the phenyl acetyl coenzyme, you can see the phenyl acetic acid here. You can see the phenyl acetic acid here. The phenyl acetyl coenzyme means what? It contains this phenyl acetic acid. This is getting attached, and the amino adipic acetic acid is getting to be replaced. The amino adipic acid which is found inside the isopenicillin get replaced with phenyl acetic acid, and this reaction is actually catalyzed by acyl transferase, right, to form penicillin G. So amino adipic acid, in short it is written here AAA. It is found inside the isopenicillin. Previously we have seen amino adipic. So this amino adipic acetic acid is going to be amino adipic acid is going to be replaced with phenyl acetic acid catalyzed by this acyl transferase enzyme, and thus we get penicillin G. Okay, so that's why it is saying that if we can modify this biosynthesis of penicillin production by introducing more genes that code for cyclase enzyme, by introducing more genes that codes for acyl transferase enzyme, it has been shown that more penicillin G production occurs. So through genetic engineering, through modification of the genes by introducing the genes that code for these two enzymes can be introduced to increase the penicillin production. Right? Now let us discuss about the production process of penicillin. Now an outline flowchart of industrial production of penicillin has been given. Okay, so let us see the outline. So we use a lyophilized culture. Well, lyophilized culture is actually a little bit of water retention culture actually, where we apply a cold temperature to freeze them and to remove the water molecules actually. So I will give you the definition of lyophilized culture in your WhatsApp group. So this lyophilized culture is actually used. Lyophilized culture is actually maintained to store the culture for many periods of time. Okay, for long periods of time. Okay, it is little bit different from the dehydration technique where we apply heat. Here we apply cool temperature, we apply freezing temperature to freeze and then to remove away the ice crystals formed in the culture. So the lyophilized culture is used as a inoculum here. I have already mentioned that inoculum is a small amount of material which consists of here the fungus to start a culture, and then this inoculum is being prepared. From this lyophilized culture we prepare inoculum, we prepare starter culture, and we try to grow them in a pre-fermenter. Well, pre-fermenter is actually it is just like a fermentation technique. It is just like a fermenter that we normally use, but it is in a small scale. It is done in a small scale. That means less quantity of nutrients are being applied here. Okay, for the growth of the inoculum. Then after suitable culture in the pre-fermenter, we grow for the fermenter where large quantities of the medium are employed, and fermenter are very large vessels as we know. These are showing the impellers which is continuously steering the culture for easily available of the nutrients to the inoculum, to available to the Penicillium chrysogenum spores. And thus after certain period of time with properly optimal temperature, pressure and aeration, because your penicillin production is an aerobic process, therefore continuous supply of oxygen will be needed in the culture medium, and temperature is maintained around 25 to 27 degrees Celsius, while the pH is maintained slightly acidic, that is around 6.5. pH is maintained around 6.5. And um, generally the medium that where the Penicillium chrysogenum are being grown in this fermenter, so that we can obtain penicillin from this culture medium. The medium which is used for the fermentation mainly consists of corn steep liquor. It mainly consists of corn steep liquor. Okay, it mainly consists of corn steep liquor obtained from the corn grain, and for carbon source we need lactose. For carbon source we need lactose to in the culture medium, Well, constrict liquor contain all the essential amino acids which are needed for the penicillin production. That is, it contains your L-cysteine, it contains your L-valine. Okay, and then after period of 150 to 180 hours, we prepare our penicillin can be extracted. It is finally filtered and after filtration we cool them in a cooling tank where downstream processes is being done. Downstream processes are mainly employed for extraction of our desired materials. Here, your desired materials is your penicillin from the culture, while the mycelium of that fungus is again used for the penicillin production. It can be again employed to the culture medium for penicillin production. So this is a little bit a brief introduction of your penicillin production in industrial process. This is enough for you to study in your examination. More details has been given. I will try to give you a separate image where more little bit details has been mentioned in that diagram. Okay, so what it is written here, let us see. So the lyophilized culture of spores, where spores are the fungal spores that we normally say, it is cultivated for the inoculum development. So lyophilized culture of spores. We have to take somewhere as a starting material. So Penicillium chrysogenum spores are being obtained from the culture, and that culture has been taken from somewhere. We have extracted that culture and we have lyophilized them. We have used cold treatment to remove away the water molecules from that culture so that it can remain for a long period of time. So that lyophilized culture of spores is cultivated for inoculum development. Just remember, spores are the reproductive parts in fungus. In case of fungus, where sporulation development occurs, and then after culturing your inoculum, it is transferred to the pre-fermenter where we grow that fungus, we grow that Penicillium chrysogenum fungus, and then to a fermenter for our final um, production. Okay, where we insert our nutrient medium for the growth of this Penicillium chrysogenum, supplemented with nutrients, oxygens. Okay, constantly impeller is there for agitation, for continuous mixing in the culture medium. Proper pH is maintained, right? So penicillin production is completely an aerobic process. So we require air for penicillin production. Well, in alcohol production, it was completely an anaerobic. So there we try to avoid the oxygen to be entered to the fermenter. But here we have to go for aerobic process. So a continuous supply of oxygen is needed to the growing culture. The aeration rate, that means at what rate oxygen should be entering, is zero point 0.5 to 1 ppm. Okay, so the aeration rate of your oxygen, that means at what amount oxygen will be entering to your culture medium for aeration, uh, for supplying air, is zero point 0.5 to 1 ppm. pH is maintained around 6.5 and temperature is maintained at 25 to 27 degree Celsius. Totally, this penicillin production is completely occurring in submerged process. That means we use a liquid medium for culturing of the penicillin. That's why we saying submerged process or submerged fermentation. Now the best medium for fermentation of Penicillium chrysogenum is your corn steep liquor. So we use corn steep liquor we of 4 to 5% dry weight, and for carbon source we use usually a lactose for the optimal growth of your Penicillium chrysogenum, because corn steep liquor contain all the essential amino acids needed for your penicillin production like L-cysteine, L-valine. Addition of yeast extract, soy meal or whey is also done for good supply of nitrogen. So for nitrogen supply we may add soy meal, we can add whey or yeast extract. Sometimes chemically we can add ammonium sulfate or um, ammonium sulfate or ammonium nitrate can also be added as a source of supply of nitrogen. Phenyl acetic acid or phenoxyacetic acid serve as a precursor for penicillin biosynthesis, which can be continuously fed. That means you can go for continuous fermentation process where continuously we will add at a suitable concentration. Obviously we will supply the phenyl acetic acid. Phenyl acetic acid serves as a precursor molecule or initiator molecule for your penicillin biosynthesis. Further, if we continuous feeding sugar, it is advantageous for good yield of penicillin. Now, some of the penicillin production profiles are given in the figure 25.4 and 25.5. Let us see the figure 25.4. There is a graph which has been mentioned here. So, if you see the graph, you can able to observe what I am trying to say. Well, on the x-axis you can see the time, and the y-axis shows the concentration of your different source which are supplied for penicillin production. You can see the highest penicillin production occurs at which region. If you see the highest penicillin production occur in that region where your concentration of your biomass refers to the Penicillium chrysogenum, okay, concentration, low concentration of ammonia, and high concentration of lactose. Sorry, not high concentration of lactose, uh, very much low concentration of lactose yield a high production of penicillin. Initially, if you see, initially we have increased lactose concentration, and slowly the lactose concentration goes on depleting as a result of batch culture. Now if we go for batch culture, that means once in a time we are adding all the things and we let the cells to divide, right? So not continuous. If you go for batch culture, see considerable period of time the lactose concentration gets decreasing because the microbes, the fungal cells are utilizing the lactose. Okay, and that's why the biomass gets on increasing. It goes to its stationary phase, as I have said in your previous class, the stationary stage of your microbes. The concentration of ammonia also gets depleted due to your utilization of ammonia by the your microorganism. Okay, so with considerable period of time after utilization of your energy, utilization of your carbon, utilization of your ammonia, you see a high increased production of your penicillin is achieved. In the next profile, if you see, the next profile shows the different type of other things which are given, like the dissolve oxygen. Okay, next the sugar feeding. The square are the sugar feeding graph, which shows the what amount of sugar we are adding, okay, during the culture period. And this is your concentration of carbon dioxide. So in the previous graph, if you see, it shows the penicillin production in relation with the substrate utilization and biomass formation. And in the second graph, it shows that during continuous culture of your penicillin, that means continuously we are supplying all the nutrients, what will be the rate of penicillin growth, which has been shown. This is shown in case of batch culture. This is shown in case of batch culture, where we can see that after depletion of the lactose, because the lactose is being utilized by the microbes, okay, with a considerable period of time, then depletion of your ammonia, we see after a period of time there will be increase in the penicillin production. And also the biomass refers to that Penicillium chrysogenum which is being used here. Well, here in the continuous culture, when we see penicillin production in relation with the continuous feeding of sugar, if we are allowing the sugar to be introduced to the medium, then what will be the concentration of penicillin produced? So you see the concentration of sugar is being used continuously in the medium at its highest point. You see suddenly after utilizing the sugar, the growth of penicillin becomes the highest. Also the other things like the concentration of carbon dioxide and the dissolved oxygen, where we are supplying oxygen to the fermenter at a rate of zero point 0.5 to 1 vvm. At a certain period of time, you see highest in the penicillin production can be achieved after utilization of all these things. Not complete utilization, because we are allowing the dissolved oxygen sugar to be introduced to the culture medium. So it is estimated that approximately 10% of the metabolized carbon contributes to the penicillin production. So out of 100%, 10% of the carbon, metabolized carbon, that means the carbon which has been used during the metabolism of penicillin production, contribute to penicillin production, while 65% is utilized towards energy supply and 25% for the growth of organisms. So out of 100% of your carbon, 10% is used for penicillin production during their metabolism of penicillin production. 25% is used for the growth of the microorganism, that is the growth of the Penicillium chrysogenum, and 65% is used for the production of energy by that microorganism. Okay. The efficiency of penicillin can be optimized by adequate supply of carbon source. So we can increase the production of penicillin by, if you can adequately supply the carbon source, right? Like by adding glucose, by adding acetic acid, we can increase the yield of penicillin by about 25%. As we can see in this continuous culture graph, that continuous supply of the sugar causes, at a certain time, there will be increase in the fold of penicillin production, you can see. For efficient synthesis of penicillin, growth of the organism from the spores must be in the loose form, not as pellets. Well, the spores are found in the form of clumps known as pellets. So you should use the spores in loose form. That's why we have to go for the pre-culture, where we have to suspend this inoculum containing spores into their loose form. Okay, growth phase is around growth phase is around 40 hours, with a doubling time of 6 to 8 hours. That means during 6 to 8 hours, there will be doubling of your Penicillium chrysogenum. After the growth phase is stabilized, penicillin production exponentially grows. So it has been shown, if you see the graph, if you see the two graphs, let's see the two graphs. You see that the biomass, that is this is the biomass in here, and the biomass here, the biomass has not been mentioned. So it is saying that when the growth of the microbes is in your stationary phase, that means what is stationary phase? I have shown to you before. Stationary phase is, wait for a minute. So stationary phase is first the microbe starts to prepare to divide. This is called lag phase. Then the microbes grow exponentially, very much in a logarithmic rate called exponential phase, and then comes the stationary phase. Okay, and then when depletion of nutrients is there, when there is absence of nutrients, then they go for death phase. So at the stationary phase, when the microbes reach here, the Penicillium chrysogenum fungus, when they reach to the stationary stage, then it is saying that the penicillin production occurs exponentially. Okay, so when the growth phase is stabilized, that means when the cells reach to their stationary phase of their cell cycle, that means the amount of nutrients present in the medium and the amount of cells are equal in number at that time. So at that time, the penicillin production occur exponentially in a logarithmic rate with appropriate culture condition. Well, you can recover the penicillin during your 150 to 180 hours. So after, during, you can increase the time, you can increase more, for more penicillin production, you can increase your time in fermentation for up to 180 hours, then you can recover your penicillin. So as your fermentation is complete, the broth containing about 1% penicillin is processed for extraction. So when your fermentation is complete, the broth, the liquid broth where you have used them to culture the Penicillium chrysogenum, and where the penicillin has also been produced by that same microbe, 1% of penicillin is processed then for extraction. Mycelium is removed by filtration. The mycelium can be used again for the next culture. Penicillin is recovered by a solvent, so we can use a solvent which will dissolve the penicillin, like you can use N-butyl acetate or methyl ketone at low temperature and at acidic pH. By this way, chemical and enzymatic degradation of penicillin can be minimized. So why we use solvent here? To dissolve the penicillin so that we can maintain its stability against the penicillinase enzyme. Okay, that's why we use this solvent called N-butyl acetate or methyl ketone at low temperature and at acidic pH, that is less than 3. So in this way we can maintain the penicillin. Penicillin containing solvent is treated with activated carbon to remove the impurities and also the pigments. Penicillin can also be recovered by adding potassium or sodium acetate. So by using these different chemicals, different chemical compounds, we can extract our penicillin from the broth medium. Potassium or sodium salts of penicillin can further processed like N-butanol or isopropanol to remove the impurities. So for removing the impurities, we can use the potassium salt, we can use sodium salts of penicillin. Okay. S- Um, okay. To remove the impurities of the penicillin. So it is showing that we use N-butanol or isopropanol to remove away the impurities found in the penicillin, and in this way we can yield the penicillin for about 90%. Okay, now these penicillin salts that are actually being prepared, by removing away the water from that penicillin salts, it become crystallized and dried under required pressure. Then that penicillin salts containing the penicillin can be further processed with some solvents, and finally we can produce the pharmaceutical dosage forms. We can prepare, manufacture antibiotics on the basis of that salts right, in a proper concentration. We also have to mention the dose also, that what amount is present in that particular pharmaceutical product. And for its stabilization, we use, I have already said, we use some solvents like n-butyl acetate or methyl ketone at low temperature, that is less than 10 degrees Celsius, and at acidic pH, that is less than 3. And further, the penicillin is treated, again I'm repeating, further the penicillin is treated with activated carbon to remove away the impurities and the pigments. Potassium or sodium salts of the penicillin that is present, it is further processed then in dry solvent by using butanol or by using isopropanol. Why? To remove away the impurities mainly. So in this way, we actually prepare the penicillin in a suitable concentration, right? Penicillin G and penicillin H are the fermented products obtained from the fungus Penicillium chrysogenum. Well, if you want to make semi-synthetic penicillin, then we have to add the precursors for the R group, right? Okay. So this was all about the penicillin production. Okay, more is information given here. Production of 6 amino penicillinic acid. What it is saying here. So I hope you have understood this part that I have mentioned with you just now, that how the penicillin is recovered. Simply, again I'm repeating actually for you to understand. So after fermentation, this penicillin is recovered by using a solvent, like we can use butyl acetate or methyl ketone to dissolve them. Okay, so that we can able to prevent the penicillin from getting degraded. Then finally, the impurities found in the penicillin is removed by using activated carbon. Okay, you can recover the penicillin from the broth by adding your potassium or sodium acetate. The penicillin salts, which are the potassium salts of penicillin or sodium salts of penicillin, they are further processed actually by using n-butanol or isopropanol to remove the impurities mainly. And finally, we remove the water from the salts of penicillin, and thus penicillin salts become crystallized, which are further at certain pressure and certain temperature, they are processed for pharmaceutical manufacture of the antibiotics, or you can say suspension is also prepared sometimes. Now, production of 6 amino penicillinic acid. Penicillin G and H are mostly used as starting material for the production of several synthetic penicillin containing the basic nucleus. Nucleus means the main head here is 6 amino penicillinic acid. So the 6 amino penicillinic acid is the main component. The R group only varies to form different types of semi-synthetic penicillin. About 10 years ago, only chemical methods were available for hydrolysis of penicillin to produce 6 amino penicillinic acid. Nowadays, enzymatic methods are preferred also for preparation of semi-synthetic penicillin. So different methods are achieved for making semi-synthetic penicillins, like immobilized penicillin amidase enzyme. That means the enzymes which has been encapsulated in a lipid layer, in such a polymer layer, where the enzyme will not get activated, but its activity will be remain for a long time. So immobilized penicillin amidase enzyme have been developed for specific hydrolysis of penicillin G and penicillin V. Penicillin salt of either G or V can be used for hydrolysis of immobilized enzyme system. pH during hydrolysis is kept around 7 to 8, and the production of 6-APA can be recovered by bringing down the pH to 4. So these all these techniques are being applied for your modification of your natural penicillin to prepare your semi-synthetic penicillins, and that's all. So just you have to need only the production process, not this one. You cannot this will not give in your syllabus. Actually, you have only been discussed about here about the production process of penicillin only. So just go for this process, you go for this process. Well, I have first introduced this process, I have first introduced that what is penicillin, and that's why the biosynthetic pathway is also an essential part. So this is also important, right? So this was all about for the penicillin production industrially. I hope you have understood this concept. And for questions, you can ask in me in the WhatsApp group.